were Excel spreadsheets. So the very first implementation of SPM was actually writing down sort of the statistical equations in an Excel spreadsheet where each cell in the Excel spreadsheet was corresponded to a voxel in order to assimilate the data in order to perform. So that was important. It's important from time to time just to forget everything you thought you knew and start again and just question everything. So at another level, it was a really important experience. Analysis at the time, it was just everybody helping everybody else. And in a particular instance, it was basically me being sent with my code on a 1/4-inch magnetic tape to Leslie Anglage and Jim Haxby and Barry Horowitz at NIH, and then copy it on there.
Welcome to Stimulating Brains.
Hello and welcome to Stimulating Brains. Today I'm deeply honored to welcome Karl Friston, one of the most influential neuroscientists of our time and a central figure in human brain mapping. It is certainly not Karl's style to boast with numbers, but I would still briefly add that his published work has been cited over 400,000 times, and he has an H-index of almost 300, which are both numbers that are typically unheard of. Many listeners will know Karl for the free energy principle, active inference, dynamic causal modeling, voxel-based morphometry, statistical parametric mapping, and many other theoretical contributions. But in this episode, I would like to take a slightly different route than featured in most episodes on other podcasts with Karl. Rather than beginning with the more modern concepts such as active inference, we will go back to the early history of SPM, or Statistical Parametric Mapping, the software and statistical framework that helped turn functional imaging from a local craft into a shared scientific language. SPM began in the PET era, while Karl was at the MRC Cyclotron Unit at Hammersmith Hospital. The first SPM software, now often called SPM Classic, was shared with the emerging functional imaging community in 1991. The first major rewrite, SPM 94, followed in 1994, and later versions carried the field through PET, fMRI, structural MRI, EEG, MEG, DCM, VBM, Bayesian modeling, and much else. So today I invited Karl to tell this story from the inside, how a psychiatrist came into brain imaging, what the first activation experiments felt like, how SPM became shared infrastructure, who the key collaborators were, how the MNI space and spatial normalization shaped the field, and how the functional imaging laboratory became a kind of training ground for modern human neuroimaging. We will even hear that the very early first version of SPM was a set of Excel spreadsheets. That surprised me. As always, thank you so much for tuning in, Stimulating Brains. I hope you enjoy the conversation as much as I did. So thank you so much, Karl, Professor Friston, to join this interview, the podcast. I know how busy you are. You even just told me that this is the third interview today. So thank you so much, even more so for taking the time to talk to us. As you may have seen or know, I always start with one icebreaker question, which is about hobbies. What do you do when not working or when not engaged in neuroscience?
I watch television.
Fantastic. Any particular shows?
Yes, I have a little routine. So as it comes to the end of the workday, my wife prepares my meal and we watch the news together. And then we watch some light entertainment or dark entertainment, depending upon the offerings on TV. And then I try and watch either a gardening program or a DIY program, and then cap it off before going to bed, usually at about sort of 1:30 with a uh, a political roundup of the day, what the papers say, and the like. So that's what I actually do. The proper answer to your question is once a year I um, take August off, and I
Mhm.
do that to commit to landscape gardening. So I just try to improve
Oh,
my little garden, building temples and pagodas and pathways and the like. And then my wife populates it with pretty plants for the rest of the year.
well, I'd love to see that. Are there any pictures of this that you could maybe share for the episode of the
Yes,
garden?
I'll send you a picture that my wife Anne took of having built a Gothic arch at the end of the garden. I will send you a picture of that just to
Fantastic.
embellish, just to prove I do things apart from work from time to time.
Fantastic. Sounds great. So, going a bit more into your career, but starting off very early, what kind of childhood or early intellectual environment shaped you? And then, maybe later, much later, who were key mentors and turning points in your career?
Yeah, this could be a long answer. Obviously,
Summarize your
when
life.
summarize my life and all the important people in it. Well, I think clearly you have to start with your parents, and I don't mean that in a trivial sense. They were extremely formative in um, determining the kind of scientist or person I was going to be and the particular sort of direction of travel in terms of scientific inquiry. So my mother was a nurse, but in those days you had to give up being a nurse once you got married. You weren't allowed to be married as a nurse, so she
Mhm.
devoted herself to bringing us up, her children. But she remained passionately interested in the way people worked and the way they behaved, and in particular popular psychology. So I was surrounded by books and conversations about popular psychology as it was um, in my formative years. My father was a bridge engineer and had a passion for physics and the like, and used to make me read another kind of book. And the one that I remember most acutely is Sir Arthur Eddington's Space, Time and Gravitation. I thought it was an eye-opening and wonderful book. Um, so I had this mixture of physics and maths and psychology. So the obvious thing I wanted to be was a mathematical psychologist when I grew up.
Mhm.
So I went to my careers advisor and said, I want to be a mathematical psychologist. And the career advisor said, oh dear, well, you need to be a doctor first, because he thought I wanted to be a psychiatrist. And I didn't know the
Mhm.
difference, and he didn't know the difference. So I spent the
Interesting.
first few years of my education being studying to become a doctor and then specializing in psychiatry. But at the earliest opportunity, try to move into research, and that's where the list of mentors arises. And the first mentor was a gentleman called Phil Cowan, who comes from a stable of very influential clinical academics in the UK that effectively established neuropharmacology as a discipline.
Wow.
And there's still, some of the good and great now. People like Dave Nutt in the UK were part of that group. That was my first exposure, which was fortuitous because that understanding the importance of neurotransmitters and pharmacology in shaping the way our functional anatomy and our computational architectures has been an enduring theme,
Yeah.
especially in relation to the next set of mentors, which would include people like Peter Liddell. So Peter Liddell was at that time becoming the good and great, but at that time, a bright young thing in schizophrenia research, and was the only person to give me a job in research, and that job was to be working with the newly formed MRC Cyclotron Unit at the Hammersmith Hospital,
Yeah.
where they just established, and when I say they, people like Terry Jones and Richard Vukovic had established the first European sort of positron emission tomography uh, um, infrastructure
Mhm.
that enabled us for the first time to look at the brain in action.
Yeah.
The very first in Europe at least, or in the UK at least, the first time to have that window on functional anatomy, to have that window on the brain at work. Um, because I was working for Peter and his special interest was schizophrenia, my job was to collect data from a cohort of people with chronic schizophrenia, which was an interesting endeavor in the sense that it is now possibly unimaginable that you would ask somebody with delusions and chronic schizophrenia to expose themselves to a radioactivity machine and have their brain scanned. At the
Sure.
same time, this was in the days when we used to administer the radioactivity, the radio traces required to form images of these tomographic reconstructions of the brain. We had to inject radio tracers and
Okay.
not into the veins, into the arteries. So this
Really?
required, yeah. Well, actually, that's not true. In order to monitor the blood levels of the radio tracers, you needed arterial stabs. So this was a sort of heroic kind of early brain imaging.
I'm
So
sure. Sounds like it, yeah.
it was. We didn't actually do arterial stabs on the patients. But one funny little story I remember from those days: a certain proportion of these, who I was also partly responsible for their clinical care, thought that the brain scanning actually cured them to a certain extent, which was very pleasing.
Very nice.
But I do remember then doing radial stabs on another great mentor, Chris Frith, who was at that time recruited to the MRC, and he was a rising star, and probably risen star in schizophrenia research, having worked with sort of structural images and discovered enlarged ventricles and the like with Tim Crow at Northwick Park. But he now moved over, and we worked together on trying to make sense of these positron emission tomography data. That was the genesis, under the guidance of Richard Verkowiak and colleagues, of the statistical parametric mapping software. So written out of need to make sense of what was at that time the biggest data available. So this was before the Human, Genome
Yeah.
Project.
Yeah,
This
yeah, yeah.
was, and we don't
Fantastic.
have.
We'll get to that, right? That's the main focus, to hear a bit about the origins of SPM and that history. I did hear you say once, probably jokingly, that you wasted 6 years of your life with the psychiatry residents. Was that a joke? Is that true? Do you think it was truly a waste given what you did later, or was it actually really important to make you know more about diseases? How would you see that?
Yeah, you clearly want me to answer the second, and you'll be absolutely right. No, it was a joke, and it
Yeah.
was it was uh, it was incredibly formative as an experience, and as you are intimating, um, it set the undertone for all subsequent developments and aspirations in subsequent research, even to this day now. You know, the software developments and all the theoretical developments that inherited from that, and in terms of just understanding how the brain work, were all in the service of trying to get a mechanistic handle on the kind of psychiatric disorders to which we were exposed as young clinicians, and in particular schizophrenia. So very informative time, and also informative from another perspective, which is probably less anticipated. When you move into this kind of field, and the field I'm referring to here is working with people with severe mental disorders in a therapeutic community, you undergo a process they call de-skilling, which basically means you've got to forget about everything you thought you knew and then reskill in this new context. In this instance, a community of about 30 people with chronic schizophrenia and their carers and their doctors and psychiatric nurses and social workers. So that was important. It's important from time to time just to forget everything you thought you knew and start again,
I like
and
that.
just
Yeah.
question everything. So at another level, it was a really important experience.
De-skilling. That's great. I have to remember that. Did you at the time already think about more mechanistic language for maybe psychiatry, such as inference, prediction, brain dynamics? or something else, given you wanted to become a mathematical
Yeah.
psychiatrist?
No, absolutely. I mean, there was always that, I realized I was on a journey. I knew I was on a journey from the age of about 15 to be a, um, what then was called a mathematical psychologist, which would now be,
Psychologist,
I
okay.
guess, called a computational neuroscientist. Um, and in my spare time, was certainly chasing those aspirations. And I can remember working out the importance of correlations or associations in associative plasticity. I thought, oh, this is clever. I'll go and see if it's an established thing. And I remember being both horrified and delighted to find that Donald Hebb had actually written down exactly this some decades later. And I was just trying to work out if I'd been born in time to actually meet him, and I hadn't. But but the horror was that I'd wasted about a year working out something that was already known. So this speaks to another I think sort of another missive or principle which I keep referring to, and more so in later life, which are one of the two things written on Feynman's blackboard, supposedly or purportedly at the time of his death. And the first one, which I'm sure we'll return to later in the context of generative models and the free energy principle, is that which I cannot create, I do not understand.
Yes,
The other one, know how to solve every problem that has been solved. So that puts a lot of pressure on
it's
making
a
sure
lot.
you
Yeah,
know what's been solved before you waste years rediscovering the same solutions. But both of those, I think, sort of bits of advice are very pertinent then and are very pertinent now, and certainly applied in those days. So yes, absolutely. Whilst caring for and learning one's clinical skills, there was always the deeper question in the back of one's mind, what's going on here? How can I get a mechanistic understanding of what's going on? How can I create a model of this? And clearly, subsequent experience in brain imaging realized that um, in a sort of pragmatic sense, in creating forward observational statistical models of the brain in
yeah,
terms of brain imaging.
makes sense. When did brain imaging begin to look like a bridge between clinical psychiatry and formal models of brain function for you? You did mention the Hammersmith cyclotron unit there. Maybe you can even expand a bit how you arrived there. You know how how that looked like at the time, software wise, imaging wise. How you maybe made that connection between the clinical work and brain imaging.
Yes, I mean, I did not make that connection. It was made for me. So this was one of those lucky accidents in one's career that you were just in the right place at the right time with outstanding questions, outstanding in many senses of the word. Um, and so I was recruited by Peter Little to work on an MRC funded project to look at patterns of brain activity in people with schizophrenia. This had been done before using glucose PET in America, but it was the first time in the UK that we'd used sort of a faster kind of water. So the key issue here was that one was able to take a sequence of PET scans, anywhere between 6 and 12 PET scans. Now that sounds, exceedingly limited from your point of view when we're talking
Yeah,
about fMRI time series with thousands of observations.
yeah,
But in those days, this was the first time you actually had a time series, a
yeah.
very, very limited time series. But it was a time series, which meant you were in the game now of being able to compare patterns of brain activation in real time under different sort of mental or cognitive brain states. So it was a really exciting time. And my brief was to use this technology to try and see if there were any characteristic differences between the patterns of brain activation in people with and without schizophrenia. But to do so, as your question invites, there were lots of things that needed to be solved.
Sure.
So the data analysis was clearly one of them. One of the key packages that was available at that time to make sense of these data was something called Analyze, spelled the American way by Rich Robb, who I subsequently met in Davos, years later.
Okay.
You know, and so that was from Johns Hopkins University, but it didn't. It was just a preparatory move to actually analyze the dynamics of that you would require to make any inferences about sort of functional anatomy in terms of which parts of the brain responded to these changes in perceptual set or cognitive processing.
Does that still have, sorry for the interruption, does that have something to do with the analyze image format, the header and image
Yes,
pairs?
yes,
Oh,
well,
okay.
that's
That was from
exactly where
that.
it came from. Yeah, yeah,
Interesting.
yeah. That was effectively designed for region of interest analysis. So if you could, you could summarize your data using a region of interest, and then that was the package of choice. But beyond that, there was nothing.
Yeah.
And the computer infrastructure we had at hand were Mac in the boxes. I don't know if that even means anything. You probably find those in computer museums now. And the most advanced, if you like, programming language we had to analyze the data were Excel spreadsheets.
Hmm.
So the very first implementation of SPM was actually writing down sort of the statistical equations in an Excel spreadsheet where each cell in the Excel spreadsheet was corresponded to a voxel in order
Oh,
to
really?
assimilate the data in order to perform a voxel or a pixel-based analysis of an authentic statistical kind. So that,
Love it.
this is what we spent my first year doing. So my job in a team of people who were all addressing really challenging problems, from the radiochemistry of keeping these machines alive and working through the
Hmm.
right through to the sort of the physics with Terry Jones, to the clinical input of patients and the relationship with the Institute of Neurology by Richard Verkowiak. David Brooks was a key player. Chris Frith, of course, had come in for to provide, and lots of other people. Everybody's from psychologists to chemists to mathematicians to engineers. They were all trying to solve all of these problems together to get
Yeah.
some one sort of sensible image out that summarized and enabled one to test hypotheses about how the brain worked. And it was a really important endeavor. And I don't want to preempt any of your further questions, but this was the first opportunity to test some sort of really fundamental hypotheses about structure-function relationships in the brain. In the sense, up until that time, all we had was effectively neuropsychology. All we had was inferences that this brain could be segregated into functionally specialized areas simply because when I damage that area, I lose this function. So this was a neuropsychological model. So the idea that you had functional segregation as a principle of brain organization was purely hypothetical. But for the first time, you could now actually look at the brain responses, the specialization in response to simple paradigms of the kind engineered by another of my mentors, Semir Zeki. What's the difference between looking at sort of black and white images versus color images? You are able now to measure in every part of the brain the responses and show, yes, the functionally specialized color center in the brain, visual area 4,
Yeah.
was responsive to color, but crucially, only that.
Yeah.
And that there was no other. So then you had a definitive test, empirical test of this hypothesis of functional segregation,
Of
functional
course.
specializations, anatomically segregated. Of course, nowadays people just take it for granted that functional
Yeah.
specialization is a
Yeah.
thing.
Yeah. Yeah.
But it was just an idea in the sort of late 80s and early 90s. And if
Just
we
based
can,
on lesional inference in a way.
absolutely,
Okay.
yeah, absolutely, yeah, yeah.
Interesting. And so for the younger listeners that grew up in the fMRI era, like myself, how did such a pet experiment look like? Maybe you can even describe this one that you just opened up with V1 or one of yours. Like you did mention you had multiple scans, 5 to 6 scans, if I remember correctly. But how did you, like, which task did people perform, or how did it work?
Well, the actual stimuli design and experimental design was almost isomorphic with the principles that you'd use in an fMRI design, in particular a block fMRI design.
Okay.
So let's just take two examples. So um, the study of functional specialization in the visual system, namely identifying things, so-called color centers, for example, would simply involve asking people for about 8 to 16 seconds to look at a particular stimulus, black and white image, during which data were acquired and then cached. And then you repeat the process, but looking at a colored image, the equivalent colored image, with all the right sort of normal controls to make sure the only difference was in the color of in this instance, the Mondrian's used to induce the responses. And then you'd rinse, wash, and repeat 3 times. So you'd have black and white color, black and white color. So you'd have 6 scans. And then you would organize them and then test the hypothesis that there was a difference, or there was a sort of a lawful trajectory over the 6 scans, up, down, up, down, up, down, and it may have been increasing with time. So you got an interaction with the order or the time, and all the normal things that one would associate with a block design fMRI experiment. So you can look at sort of early PET scans as basically lumping together sequences of blocks in a block design fMRI experiment, just giving you, sometimes 6, but later on 12 bites of the apple. So conceptually not dissimilar from fMRI, but fMRI was 2 or 3 years away. So a lot of the problems that would subsequently be required to be resolved for fMRI were actually addressed in the context of PET, such as spatial normalization. If you want to leverage the signal to noise reduction inherent in averaging, or put another way, if you wanted to demonstrate that this particular aspect of functional anatomy was conserved over subjects, then you had to put them into the same anatomical space.
Yes.
So this induced the notion of spatial normalization. Not only that, but you also had to register the PET scans in case the people move. And of course, if you've got chronic schizophrenia, you're likely to not be able to sort of stay still. So the things like spatial registration, spatial normalization, they were all open problems that required solutions, solutions that in spirit still survive today in the software and in the sort of state-of-the-art techniques that people bring to the table. Even if in implementational detail they have obviously improved. But these things were solved. What were not solved when fMRI came along was the very fact that you can now acquire an image in a matter of seconds with a TR of say 2 seconds or even less nowadays. Then that introduced something that wasn't in the PET data. In the PET data, the pulsar emission tomography data, you were taking data samples minutes apart because you had to let the radioactivity wear off before you then started to inhale the radioactive oxygen that was then labeling your playing the role of a radio tracer that could be picked up by the scanner. But when you're acquiring data generated through exactly the same hemodynamic and neurovascular mechanisms that fMRI, or bold fMRI at least, rests upon, you're now acquiring data faster than the natural correlations in the data. So then there's a whole other period of development, not only having to deal with the spatial correlations induced by working with extended images, and then that's another story which underwrites the development of statistical parametric mapping, but also the correlation structures and the introduction of sort of convolution models and deconvolution implicit in the inversion of convolution models apt for fMRI data.
And with that, maybe for the listeners, you mean the HRF convolving a stick model with something like
Absolutely.
HRF, right?
Yeah,
So
absolutely.
so the hemodynamic response function, how was that discovered? I mean, we're jumping to fMRI pretty quickly now, but that's when came that canonical HRF function about? Was that
Well,
early days? Like,
I mean, yeah, it's a great question because it inherited exactly from how to separate fMRI signal in fMRI time series from noisy, smooth fluctuations. And this is not a trivial problem. And I remember people like Ed Bullmore spending many years of their life with colleagues at King's looking at this, as did we. So to do this separation in this particularly difficult problem that your random effects or your random fluctuations are not identical between the observations. So if they were identical, you could just use standard least squares estimators and standard parametric statistics. But when you've got serially correlated data, that becomes much more problematic in terms of the effective degrees of freedom in time. How many independent observations do I actually have in a sequence of fMRI data that have been sampled so quickly that the noise or the random fluctuations at least are contaminating each successive acquisition slice, for example. Um, and that really rests upon understanding the dynamics of the signal. And understanding the dynamics of the signal can go in many different ways, but mathematically you have to do it. That which I cannot create, I do not understand. So you have to have a mathematical model of the fluctuations that are induced by experimental design, the effects of interest relative to the random fluctuations that are not. And therefore, we need to understand neurovascular coupling. So now you need a model, a generative model of the way in which neural activity causes what you observe, the bull signal, which is through a hemodynamic response. That's a dynamical thing. There were at that time hemodynamic models that would link blood flow to bold, known as balloon or Winterkessel models, um, um, pioneered by people like Richard Buxton and Mandeville and other colleagues. But there wasn't a full hemodynamic model that ran from neural activity induced experimentally through to the hemodynamic response. So we had to build these models. And when building these state-space models or differential equations, if you pinged these things, then they had the functional form of a hemodynamic response function. And a first-order approximation to these usually nonlinear responses, because these hemodynamic models have an inherent nonlinearity, say due to the elasticity of the vascular architecture and the like. But to first order, you could then treat this as a response function, and it would be the hemodynamic response function, and it became the canonical hemodynamic response function. Simply because it was, its functional form was so conserved over brain areas and over people in humans. And that in turn, well, how do you put that into a model? And you can go one of two ways there. You can sort of remain in the world of differential equations, state-space modeling, and that ultimately became known as dynamic causal modeling.
Yeah.
Or you could say, okay, let's just take a first order approximation and put it into a general linear model. How do you do that? You do exactly what you just said a few minutes ago.
The
You
stick
just
function, yeah.
Exactly. So your stick function now stands into the sort of neuronal perturbation, the
Yeah.
experimental effect that you've induced by experimental design. And when you convolve with a canonical hemodynamic response function, you're now basically replicating what you would have done if you'd integrated all the dynamics and the neurovascular coupling in a full state-space model or dynamic causal model or the same kind of thing. And that then became known as a sort of the classical GLM, but it's actually a convolution. Because you're convolving your stick functions, you're actually creating a good model of the hemodynamic response to a neuronal perturbation or input. But the story doesn't end there, of course, because just because you've now got your a good model of your signal, you still have to contend with the correlations amongst the noise the random fluctuations, which now led to the notion of the effective degrees of freedom. Um, and that required a reapplication of the theory of stochastic processes that have these correlated structures in them that would used to do the spatial correction, effectively looking at spatial degrees of freedom when making inferences in statistical parametric maps, now had to be applied in the context of the temporal behavior of fMRI time series. So there was an interesting sort of repurposing of the maths that we developed for dealing with spatial correlations, spatial smoothness, to handle temporal or serial smoothness in fMRI time series.
Interesting. Very, it got complex pretty quickly. And I guess deconvolving from bold signal back to neural space is probably even more complicated because there's not just one solution, I assume. Is that
Absolutely? Yeah, no,
okay?
yeah. I mean, traditionally, I mean, it's not something that you need to do unless you actually need an estimate of the underlying neuronal response
Yeah.
to, for example, construct psychophysiological interactions. But
Yes, yes,
generally
yes. That's
speaking,
what. Yeah.
yeah, you don't need to do that. If you did, you classically you'd appeal to something called vena filtering. The problem is vena filtering, you're normally assuming serially uncorrelated errors. However, it's not really a problem in the general
Yeah.
spirit of things because when you invert a general linear convolution model, you are doing the deconvolution. So you don't need to actually estimate the neuronal response. All you need to do, again, referring to this notion of the importance of a generative model underneath the data,
Yeah.
in the spirit of that which I cannot create, I do not understand. You
Yeah.
actually have to build something,
You,
a model of it before you
yeah.
understand it.
You
But
build the,
inverted,
yeah, makes sense. I knew the deconvolution from PPIs as well. So I know that in the general task-based fMRI analysis in the GLM sense, you don't need to deconvolve. You construct based on the neural behavioral or perturbational task data. You essentially build a fake bold signal, right? An
yes,
estimated bold signal, and then you compare that with every voxel in the brain, right? In with the real true bold signal and get a beta estimate. That's my basic understanding of the idea, which I still find very brilliant. And you did mention, when we talked about the PET data before, the control condition, right? I think the general idea of doing something like this even stems from Donders back in the day to have kind of two different contrasts or conditions that are almost the same, but there's one thing that's different, right? And then you
yeah.
subtract them from each other. So I guess that is probably one of the big principles that, I don't know if you had to invent it or you had to come up with to even make sense of PET and fMRI data. Is that right?
Yeah. Again, that's a really insightful question. Before I answer that, and just tell you little stories about how we address that and how we and others address that. I just want to emphasize your important observation that having a forward model, a generative model, a model that maps from causes to observable consequences, um, if that model is a convolution model, then inverting or fitting that model is a deconvolution operator. So that's what I meant by, we're always deconvolving, even in our
Okay.
perceptual synthesis,
Yeah.
or e.g. reconstruction or fitting fMRI linear convolution models. These are all deconvolutions, but they're deconvolutions simply because we are inverting or fitting a convolution model. So that's why you don't need to do the deconvolution explicitly. You just need to invert the right kind of generative model. And in this instance, it was a convolution model. That's such a general principle which has stuck with me and I think all of my colleagues throughout the decades. I think it's worthwhile saying out loud and using your words. But the other fascinating, yes, you're absolutely right. And of course, we were not alone in this. So we're talking about the nascent field of human brain mapping as we know and love it, pioneered by work by people like Mark Rakel and Peter Fox and Steve Peterson and other colleagues at WashU and St. Louis, and lots of other people around the world. Alan Ems's group in Montreal. And well, I won't list because the people will be upset if I don't list them. But just to say, there was there was a community that led to the organisation of human brain mapping, because before that, the only place that we could talk to each other was the Society for Regional Blood Flow and Metabolism. So, commonly called the gerbil stranglers, because they were interested in stroke. They were interested in understanding hemodynamics as it pertained to pathology and the consequences of neurovascular accidents or stroke. So their focus was not really on the big questions, how does the brain work?
Yeah.
It was really understanding how perfused brain tissue would respond with differences in the neurovascular architecture and lesions to it. So a lot of these people initially got together to talk about these problems at the Society for Regional Blood Flow and Cerebral Blood Flow and Metabolism. And it was only after a few years that the Organization for Human Brain Mapping was formed as quite a small society, just to allow people to shout about these issues. And the key issues, of course, at that time were experimental design. And you're absolutely right that sort of the very early subtraction studies did appeal to Donda's notion of cognitive subtraction, which you could think of as the very first sort of brain mapping experiments, in the sense that he was looking at the temperature of the skull that was overlying the functionally specialized responses with and without ringing bells, for example. So, so immediately that started to lead to a more principled approach to experimental design and the ontology of designs that survives today as the basis of a good experimental design. And I should add, sometimes violated by sort of new wave approaches as each generation comes and learns the skills of the trade. But eventually they will all return to this kind of ontology of experimental design. And the first important move is to move beyond simple subtraction designs, where you've got one activation condition and one baseline, and you're just basically doing a careful task analysis in order to identify the one task component or processing component that differentiates between these two conditions, and then you can attribute the difference in activation to this particular task component. What becomes more interesting is when you now turn to factorial designs, when you've got two factors. Because if you are making the assumption of what's called pure insertion, which is where most of the original debates and arguments and advances, largely due to Kathy Price, who then became sort of famous in language and recovery of stroke research. If you now look at factorial designs, then you have to immediately develop a sort of a mental image of differences and differences and differences, which would be the interactions, and the importance of making sure that your experimental design is balanced in order to efficiently estimate the interactions to test the assumption of pure insertion. And by pure insertion, what I mean is, if I add this cognitive process, this task component, to this compound task, is the extra activity that I see purely explained by the addition of this component, or have I changed the context in some way, and there's an interaction now between the added component and the task at hand? And the only way to really address that is to have a factorial design. So that was the rise of factorial designs. And then you ask yourself, well, what happens if one of the factors is not easily binned into categorical things like sort of black and white versus color, or rewarded versus unrewarded? What happens if it's more subtle, like the amount of reward, or the brightness of an image? And then you move into parametric designs. So you've got these sort of two moves you can make. You've got sort of your standard subtractive design that can then be embellished with multiple factors to multifactorial design, and any one of those factors can now be made parametric. And all of this had to go into the general linear convolution model. So, immediately you move, if you're just dealing with PEP, from an analysis of variance kind of models with indicator variables saying that this is condition one, this is condition two, this is condition three, to analysis of covariance, where you're allowed now for parametric regressors and their interactions. So, to summarize a long response to your important question, there was this emergence of not only the conceptual ontology that you brought when trying to optimize your experimental design, but also the software under the hood that would enable you to actually use the data to address the hypotheses that were implicit in your experimental design.
So I guess Excel was not good enough anymore at that point. What was next there?
Yeah, so we at this point we've moved to MATLAB. Yeah, absolutely. So
Why MATLAB? What was it's interesting now with all the Python buzz. I'm a dinosaur already. I still work with MATLAB quite a lot, but I remember loosely this used to be a academic solution back in the day from was called Matrix Lab and was then bought by MathWorks, I think. Was that a deliberate choice? Why MATLAB? Was it the only powerful enough tool that could handle this, or how did you choose that?
Yeah, yes, that is, I think, a very pressing question in relation to the current celebration of the community that you can now do SPM in Python, which seems to please a lot of millennials. But like you, I can't quite get into the joy of that because I still work with MATLAB. But you're absolutely right. At that time, MATLAB was the academic software. And actually, I might argue it remains the academic software for true academics, not in machine learning, and not in getting your chatbot to write code for you. You'd have to go to Python for that. But when it comes to sort of high-end academic inquiry, then MATLAB was seen, and still to a certain extent. I think now sort of software engineers, I think about sort of 56% use Python and only about 3 to 4% use MATLAB anymore. But in academia, I think you'd probably find many more people use MATLAB. And certainly at that time, it would have been 100%. So why MATLAB? Well, it was a third generation or third order kind of language. So it meant you didn't have to learn C or C++. You could actually get straight in there. And crucially, you could write down the expressions that you would find in a statistical text on how to analyze your data. So you could write down literally the expressions you would find in a text. Wikipedia didn't exist in those days, but standard text on general linear model, for example. So it was this very high order language that allowed you to express code in a way that was readable by somebody else in reference to their understanding of what they were creating as their generative model. So it was pedagogically a really important move. So we certainly did not want to write it. We could have written it in C, and it would have been much, much more fast and much more efficient and much easier to use. But it would have had absolutely zero educational, or so it would not have been useful in socializing the ideas and explaining what they're actually doing under the hood. And it's interesting, I think, you sort of bring up the pedigree of MATLAB. So before it was commercial, you're absolutely right. It was written by people like you and me in the early days of X-ray crystallography. So I think in New York, I may be wrong, but people with our academic aspirations and funding, were suddenly confronted with this massive X-ray crystallography data. How on earth do you make sense of this? And what they needed, of course, was some way of doing really fast Fourier transforms, not dissimilar to sort of tomographic image reconstruction in positron emission tomography, and indeed sort of fMRI image reconstruction. So they actually built this software that was super fast and really accessible for fast Fourier transforms and handling large matrices, which was, of course, perfect for brain imaging that had
Sure.
very large matrices. So it was a natural choice, and it was in the spirit. We were using MATLAB, and I don't know that we even had to pay for it initially. It was academic software, and I think I can't remember. This is probably a made memory or false memory, but I think the notion of paying for it or paying for a license only came in a few years later,
Makes sense.
where people realised that suddenly there was an uptake in this community, and indeed nowadays, literally this month, MATLAB have started to fund. Or look at funding SPM development because it is still used and written in MATLAB, although now it can be transcribed into
Sure.
Python.
Interesting. Okay. And I know you are pretty humble, so you probably will not prefer to speak too much about yourself, but was it truly you that wrote the first version of this, or was it always a team effort? Because I mean, you were the main person behind the toolbox. Everybody knows that, but who else played a role? And then how did I assume it was probably small, humble beginnings, mainly written for yourself or for the internal use, or how did that all come about?
No, that is absolutely right. It was just small internal use, and literally small, with small Excel spreadsheets and small Mac-in-the box computers
Yeah.
for one or two other people. Long-term friends, people like David Brooks and colleagues in Belgium trying to analyze their. Actually, this was receptor radioligand binding studies, looking at Parkinson's disease. And then when handling larger data of the sort required to analyze the schizophrenia data that we were required then to analyze, we then moved to MATLAB and a more expressive environment. And you're absolutely right. It was just a handful of people who were rushing to ensure that there was a pipeline that could make the optimal sense of these data by sticking to first principle accounts, and good practice in terms of a generative model and sort of Bayes optimal or maximum likelihood optimal in its original instantiation solutions or inversions of these generative models. So Andrew Holmes was a really key player in those days. He subsequently got seduced into pharma and industry, and is probably very, very rich somewhere in the north of England. But originally, he was inspirational in terms of co-writing the code and making sure that the implementation of the general linear models that were under the hood in those early code bases was correct. Jean-Baptiste Pauline was a visiting scholar from Paris, and I think he's currently in San Francisco area, but spent a lot of time in Montreal as well. He was, a close friend and hill walker with Andrew, and together they'd sort of potter around and embellish stuff. John Ashburner subsequently got into the game. He was actually a chemist originally, but got seduced into the computer science and the spatial normalization. About a year or so after the sort of Excel level sort of implementation of spatial normalization. So he was another key player. So all of these characters, with the exception of Andrew Holmes, are still on the scene. Some of them sort of pursuing a sort of open science education pedagogical role. Some still deeply committed to generative modeling and computational anatomy, like John Ashburner, and me still sort of carrying the torch.
And John Heather was another name I read.
Yes.
Is that right?
Yes, I forgot to mention John Heather. Yes, absolutely. Where did you get that from?
I'm not even sure. I think I don't know if it was from the SPM book or from, I have that in my cupboard actually, the old, red one, or from the website. I'm not sure, but I have it in my notes here.
So I forgot about John. Yeah, so John was, he was a sort of what would be called a data scientist nowadays, but a sort of software engineer, systems administrator. No, he was a more sort of paternal figure to all these young 20-year-olds who were actually writing software at that time. I think he ended up in Thailand, sort of having a nice time. Uh, I haven't heard from
Interesting.
him for decades.
John Ashburn, in my naive view, looking back, is always, I associate him mainly with the spatial things, right? Normalization,
Yes,
co-registration. Is that Dartel in the
that's right.
end, or shoot
Yeah.
and before that segment? Is that a unified segmentation? I mean, is that accurate or
Yes, that's what I meant by the computational neuroanatomy. Yes, he was
yeah,
interested in the spatial transformations, and as you say, that list of subsequent developments were all refinements of the way that you would generate an image of a particular person's brain. So again, under the hood of all of those developments that John has been pursuing over the decades, is a commitment to understanding or normalizing, registering, classifying, segmenting a brain that has come from this person by generating that particular person's brain by starting with a generative model that is a canonical brain and then warping it in the right kind of way
yeah,
by physically plausible way until it matches the observed brain. And
yeah,
then because it's this notion that you can do deconvolution by inverting a convolution model. In this instance, you can unwarp a brain by inverting a warping model. So if you start from a
makes sense.
standard atlas, for example, as supplied by the work of people like Alan Evans, the MNI
Yeah,
as standard atlas, and then warp it in the right kind of way using things like Dartel and Shoot and further more biologically plausible ways of shaping and warping brains to make it match this person's brain, then you've solved the problem, provided you can do the inversion.
yeah, interesting. Makes sense, and we use that every day, of course. In the PET era, you probably did linear registrations, right? There's not enough contrast.
Well, that's certainly true for most applications, and however, the early sort of application of this sort of generative modeling approach, sort of taking a template and warping it, did actually use um, nonlinear nonlinear basis functions, but they were very low order.
Mhm.
The reason that I may falsely remember that is that when we came to spatially normalize the schizophrenic data, the PET data from the people with schizophrenia, there was an enormous variability of a nonlinear sort in
Mhm.
terms of different patients sometimes having particularly enlarged ventricles or cortical thinning.
Sure.
So it was a very heterogeneous, and we deliberately wanted that. We wanted that within cohort variability to look at sort of parametric effects and correlates
Yeah.
within the syndrome of schizophrenia, not schizophreniform disorders. Um, and so we actually did need to use nonlinear, but the degree of nonlinearity is easily tailored by the number of spatial basis functions that we used in order to do the warping. So the warping fields can be very, very smooth, and in those days, a linear mixture of low order spatial basis functions, things like discrete cosine sets,
Yeah.
and you can truncate it to control the degree of smoothness. So yeah, um, a lot of the very earliest spatial normalization was literally an affine normalization, a linear 12
Yeah.
parameter based upon landmarks identified on the ACPC line, which was a gift to people like Peter Fox and the St. Louis group.
Yeah.
But spatial normalization, proper in the sense we're talking about it, started off gently nonlinear and has become increasingly nonlinear and
Yeah.
accurate and more precise as the years have rolled on.
And the ACPC registration has, of course, a deep history in stereotactic surgery, right? Which, this podcast is often about the brain stimulation and the sorts. There was the Talairac Atlas originally, I think, created for clinical purposes, but that became a first space, right? Before the
Yeah,
MNI space.
absolutely. Yeah, yeah. So that's, I mean, that nods to the heritage, so the clinical heritage of cerebral blood flow metabolism and its society and all the clinicians who were involved. So you had physicians, but you also had surgeons.
Yeah.
So, that was and remains, I imagine, the reference brain for human brain mapping. And so I mentioned the MNI space, which was basically the Montreal's version of a Talairac frame of
Yeah.
reference. that, as you say, is and was predicated on the AC anterior commissure posterior commissure line. And I imagine most of functional neurosurgery nowadays uses exactly the same reference plane, and which becomes particularly acute when it comes to all the subcortical machinations that you love.
Yeah, I mean, it is interesting to me, because I kind of came in my, comparably young history. I came from the human brain mapping field and then kind of traversed into the more clinical realm and became interested in the ACPC space. So I kind of learned about the MNI space first, and then the functional coordinates, which are simpler, which are really just 3 landmark points in the brain, and then relative to that, a normal Euclidean coordinate system. And I always thought the nonlinear form of an MNI transform makes more sense. It would make things more comparable, would make things maybe more precise. And I think as time goes on, there are some targets. For example, Helen Mayberg's target for depression in the subgenual cingulate is just too far away from the ACPC, or too variable regards to a linear ACPC transform, that they use more and more the MNI coordinates. But much of the, say, subcortical, subthalamic the brain simulation often still reports standard ACPC coordinates where 15 millimeters could sometimes be lateral or sometimes medial to the nucleus because there's no correction for it. And I think it's coming more and more also to the clinical field to use these more nonlinear transforms where a coordinate is transferable across brains, right? Where
yes.
So, but it up till this day, it's not always being used, and surgeons sometimes mistrust nonlinear transforms.
Oh, I didn't know that. Right. I mean, from my perspective, or imagine the perspective of people like Alan Evans and his colleagues, they attempted basically just to get a group of spatially registered neurotypical images, structural images, into the Talarak space. So that that whole point, that the whole exercise was to basically provide an image-based, a pixel or voxel-based atlas that would go hand in hand with the Talarak atlas per se, based upon a single individual, but sort of conceptually committing to exactly the same frame of reference and the same origins. And from our point of view, in terms of spatial normalization using nonlinear deformation fields, I think Mike Miller was also working along these lines. It's a long time ago now. Fred Buckstein. Anyway, these are names which it would be courteous to mention, but I may be mismentioning them. The agenda was basically to get every brain into this Talarak space that was nuanced through the MNI averaging. So there was a linear affine transformation from the MNI space to the Talarak space as defined by the Talarak atlas, which some people worried about and some people didn't. But the whole point was to get this person's brain through a nonlinear, inverting the nonlinear warp into this linear space, and that's exactly the unwarping that was solved by starting with the Talarak space as embodied in the Talarak, in the MNI's brains, as the generative model for the spatial normalization. So, it's a shame that people are suspicious of the nonlinearities, because the whole point of spatial normalization is to remove the nonlinearities to get you back into a nice ACPC frame of reference.
And I guess it's a matter of training, right? And I mean, surgeons might be, worried for good reasons, because of course these transforms can be misleading and wrong sometimes, if it's about precision in millimeters. But my perception is that younger neurosurgeons that have trained with these techniques and done them themselves are less worried, or more, it's as always, it's a tool, right? You can use it correctly and wrong. But back to the history here. So when did the MNI space come in? You mentioned Alan Evans, and MNI 305 space was the first one. Is that correct? Or
yes, I
that
think
was
that's
a
right.
linear average, if I remember.
Yes.
Right? Okay.
Yes. Yeah.
And then the more famous one, which had 152 nonlinear warps as well. I think there's a linear version and a nonlinear version. How did that? It's also interesting in terms of just that must have been a time when SPM was already a thing, probably used worldwide. When you started collaborations with Canada, how did that? Who were the players? How did that come about?
Well, the spatial, the commitment to those particular spatial coordinates, that was, it was multilateral. Yeah, there are lots of players. We mentioned the St. Louis group, the Los Angeles group, John Mazziotta's team, Art
Mm-hmm.
Toga and the like, and all the work being done by all their colleagues. And Roger Woods, I think, was a player, had something called ART. I can't remember
Okay.
what the acronym stands for. And then you have the NIH people,
Yeah.
sort of Jim Haxby as a young man, Leslie Ungerleider, and subsequently people like Peter Bandettini,
Yeah.
joining them. So these were, and many, many others, but not that many. You could fit them all into a large room.
Mm-hmm.
Were the players, and I think there was a joint commitment to this particular space, and we all used the hard work done by the Montreal group in creating these disseminable atlases, averages that serve the role as templates, if you like, for this generative modeling or spatial normalization. And I should say that uptake was not just in terms of data analysis, but sort of also in establishing the very first examples of open science databases. You could argue that SPM was the first example of an open science data analysis software.
Mm-hmm.
But in terms of databases per se of the kind that were emerging at that time under the auspices of the Human Genome Project, the Human Brain Project, no, not the Human Brain Project, the Peter Fox's initiative to try and now collate the accumulating evidence for different kinds of functional specialization in an atlas format required a commitment to a particular standard reference frame, and that
Yeah.
was the M&I version of the Telrac space. So he played an enormous role having, remember, not monthly, yearly meetings in San Antonio where we'd all gather and talk about how to standardize things, because these were also questions that the later cerebral blood flow metabolism meetings and the very, very early organization of human brain mapping meetings. So it was a general consensus among not a necessarily massive number of people. And this was a small field.
Yeah.
You couldn't really afford to do PET, and no one really had easy access to fMRI at this stage. And if you
Sure.
did, you had to pay a lot of money for it.
Makes sense.
So, it wasn't difficult to reach consensus. It was all about trying to present ourselves as a coherent collegiate community with valid internal standards. That was interestingly quite important at that stage. And I think people like Peter Fox recognized this, and I only post hoc really recognized it, because there was a lot of competition from other fields.
Mm.
And sort of one interesting sort of perspective and story I remember from that era, you know, when those very early PET studies came out, everybody, it would look like from a retrospective position that you were just picking the low-hanging fruit. At the time, it was pioneering work, the
Yes.
first proof of functional specialization in the brain. So from our point of view, from our naive prospective point of view, this was cutting edge science, and because of that, it was, one had the audacity to submit everything to nature, and of course, nature accepted everything. So, from the point of view, so it was quite normal for me to have my weekly. Well, that's not true. I only ever had one nature paper, but it was quite
Yeah.
normal for the community to have their weekly nature or science paper. That was the way that science worked. We were
Yeah.
doing
Yeah.
cutting
Yeah.
edge science, but of course, if you'd spent decades in a monkey electrophysiology trying to address similar questions, training your students to keep monkeys alive and do invasive electrophysiology and all of that, you were horrified because your experiments would take 2 to 3 years to
Mhm.
train the monkey to do the recordings, to analyze the data. You'd have 2 or 3 monkeys at most. 2 or 3 later, you write your Nature of Science paper. But we were doing it every week. So, because
Yeah.
it's so easy to acquire the data. So there's a
Mm.
lot of under the hood angst from non-brain imaging community, neuroscience,
Interesting.
consistent neuroscience community, about the potential hype of brain mapping at that time. So there was a lot of pressure on us to establish our rigor and I repeat, sort of that we're using standardized and techniques and well-validated good practice. And part of that involved a common commitment to standard spaces and interoperability
Of
and
course.
communication. And part of that was all committing to the same canonical space within which to report our results. Of course, the other part was that the really detailed work done with people like Keith Worsley and Andrew Holmes on the correction for multiple comparisons, because that was a really big thing.
Of
If we
course.
didn't get that right, then we could be accused of all the detractors of brain imaging at the time, which were numerous, ranging from people quietly muttering in their neurophysiology labs to people like Fodor, big people and the philosophy of science, for example, possibly even philosophy of mind, critiquing brain imaging.
Yeah.
And lots of arguments about false positives and
Yeah. Yeah. Yeah.
the lack of statistical validity. So all these things had to be countered. And that meant that, unlike nowadays, I have to say, there was a lot of pressure on being statistically extremely valid and rigorous in the way that
Mhm.
you reported your results. You couldn't say, oh, this looks like the default mode, and I've done a bit of representation of similarity analysis, and this looks a bit similar to that. You had to be very, very, very precise in terms of protecting yourself against false positives, simply because any excuse that a peer reviewer from outside the field had to reject you, they were going to use because they saw you sucking up all their resources, all their space in nature, all their PhD students. You were you
Yeah.
were you were basically a competitor.
So it was not, even though there were weekly papers, it was not easy, right? And it was still hard work, I'm sure. My doctoral father, Felix Blankenburg, who trained with you back in the day, I'm sure you remember him. He once
Absolutely.
said, this was my early days, so I hope it's correct, that he said you sometimes dictated papers. Is that true?
Well, but by the time Felix was intimate enough with me to know that was absolutely true. Yes, yes.
That's very impressive. Yeah, to have the physical or the mental capability of dictating it. Yeah, he's
Oh, well, if
interesting.
you don't do that, I would certainly try it. I find that you're much more coherent and natural when you're actually just speaking to something or
Hmm.
somebody. So if you pretend you're speaking to somebody, try to explain it to your mother or to your student, the actual narrative that comes out and the structure of the text is
Yeah,
actually much more convincing. I find I write, if I want a really important paragraph to look good, I could type it on the keyboard in a few seconds, but I don't. I deliberately get up, walk around with a dictaphone because I have to think about what I'm saying.
yeah,
Yeah. So
makes
it's
sense.
not,
I heard it before my first paper, so back then it wouldn't have been an option to try and dictate. But now you're right. I should maybe try it. Yeah, that's a good point. So one other question, was there ever an SPM1, or is the early sequence better understood as SPM Classic, I think, and then 91, 94, 95, 96, 99, and so on?
I think so. Yes. Again, you probably best ask people like John Ashburn, who are slightly younger than I am. I'm getting to an age where most of my memories are probably made memories. But I think that's absolutely right.
Yeah.
The SPM Classic was just the code that we wrote in MATLAB that we gave to friends. And,
Hmm.
you know, if I was going to say if you remember, because you could remember, because you were probably still at school, if indeed if you were born at that stage. Um, so this was in the era before email.
Yeah.
It was and certainly before social media, but certainly even before email. So this was in the era when you wrote a paper, you couldn't email your paper PDF format to the editors. You couldn't use PDFs and computer graphics to make your figures. You had to sort of cut out photographs and paste them on. And I've still got some early examples of the very first
Wow.
spatial organization papers that we used. And then they would have to be hand typed, and then assembled and then sent by snail mail to the editors. The same with software. You couldn't electronically communicate software, so
Mm.
that meant physically carrying. In those days, it was
Floppy
quarter-inch
disk.
magnetic tapes.
Oh, okay.
So the original, and it sounds like a grand move nowadays. And of course, in retrospect, people will tell a story that this was the first commitment to open science, and it sort of subverted the commercialization of data analysis software, and it was the right way to socialize um, and democratize data analysis. At the time, it was just everybody helping everybody else.
Yeah.
And in this particular instance, it was basically me being sent with my code on a quarter-inch magnetic tape to Leslie Anglade and Jim Haxby and Barry Horowitz at NIH, and then copy it on their computer and see if they can get it to work too. So
Wow.
this was in the days before people like Bob Cox and Adney. It was really we need some software to analyze these data because we don't know what to do with it. Carl,
Yeah.
can you send Richard? Can you send Carl across? See what he's got.
Okay.
So
So
I had a lovely
you were in
time.
Richard Krakowiak's lab when you really like built SPM at the time. That was the
Yes, now
main.
Richard
Okay.
Kowiak should be, I think, sort of acknowledged as the group leader that
Mhm.
was in charge when all of these innovations and developments were first conceived of and prosecuted. Absolutely, yeah.
And then when was so you mentioned the first one was just given to friends. Was the first version that was maybe more meant as a real toolbox than SPM 91, and was that yeah, like was that the first time people outside your immediate list of collaborators could find it somewhere openly, or it was accessible, or when did it become maybe, yeah, with the internet probably only, right? Or when did it become
Yes.
a thing?
Yes, I can't remember, but I certainly do remember what 1 year living in the basement of Jim Haxby's house for 2 weeks, teaching them how to do SPM analyses, and next year answering questions on the SPM helpline from everybody around the world. Yeah, I
So it
don't
did
know
take
how that
off at some point.
happened.
Yeah,
Yeah,
okay.
I can't remember. You don't. You have to ask John Ashburn. I think he'd remember better because, as I say, he loved at that stage the computer science, um, and he'll know who made that happen.
Yeah,
Possibly
yeah.
Jean Baptiste.
There's there's tons more questions I would have, but in the interest of time, I typically stop with rapid fire questions to just round up the conversation. There's one last question that is more individual, which is just this legacy. Maybe you can reflect on that a bit. When I started in the field in Germany, most of my mentors or the level, one generation above me were almost all trained at the field. So everybody with a name was there, had been there. I mentioned Felix Blankenburg. There's so many, like Cornelius Weiler was my other doctoral father in Freiburg, and then Stefan Klöppel in Freiburg, Christian Büchel. But there were so many more. I'm just listing a few random names here. So at the time, really, the Phil was, and I mean, it's still one of the most important centers, but I think at the time it was really the cradle of fMRI analysis with, of course, some other centers like Martino's maybe and WashU, and you mentioned a few, but how did that maybe feel also when then becoming older and just seeing everybody thrive and kind of these spin-offs and having created or co-created such a field? Can you reflect a bit on that?
Well, it is now, in retrospect, immense pride, but at the time, it was just the natural way of things. And I think you're absolutely right that it emerged. Perhaps just to put things in context, much of what we were talking about started at the MRC Cyclotron unit in Hammersmith. And then a few years later, during which time I took a sabbatical with Jerry Edelman at the Neurosciences Institute in America, but then returned, and within months we moved into the FIL, the Functional Imaging Laboratory,
Yeah.
under the auspices of the Institute of Neurology, that subsequently then was taken over by University College London. Um, so the FIL that you're talking about, the sort of the longer period under again the auspices of Richard Verkowiak, when it became established as a world centre in brain imaging and image analysis and modelling of that particular kind. That was absolutely its heyday, and it it started off exactly as you say, in terms of just training up the next generation of people to use the software that was currently being developed.
Mm.
Um, but after a few years, that just became a habit. And I do remember saying our job is this basically as a finishing school. So we used to take
Yeah.
the brightest young people, usually from Germany or France. And I remember all the Germans, there were some excellent Germans there. And we just took the brightest young, sometimes actually clinically, a surprising number of clinical young
Mm.
theoreticians
Yeah.
or psychologists. And they weren't necessarily sort of mathematicians or statisticians. They were sort of, from the sort of human sciences and clinical sciences. And we were a finishing school. So we expected people to come in, do 3 years, probably at most. Sometimes people hung around for 5 years, and very, very occasionally one of them stayed and then became a senior. But it was quite a small unit, in small
Yeah.
groups. So there wasn't really, we weren't really trying to sort of build a little empire.
Yeah.
We saw ourselves really as a finishing school.
Yeah.
And had great fun doing it because at the time all these people, Felix and Christian and colleagues were coming through. All the issues that was confronting these young scientists had to be solved, and so we were every day solving one problem or another problem, instantiating that in the software, hence the from 91, 90, 94, whatever, and at the same time skilling these people, but also learning from these people through their problem solving, and using the questions and the problems they posed as a focus of direction of travel on what should be developed. So you're always responding to needs. It's always a game of how do you best enable. And you best enable by teaching and disseminating and responding to
Yeah.
questions and needs. And that just became a sort of didactic pedagogical exercise. And that was the fill for decades.
Yeah.
We were a paper mill.
Yes.
A little sort of we've been called the paper mill. Some other lots of other sort of neuro Disney sort of things that people go for a bit of
I
fun
love that.
for a few years.
Euro Disney. Yeah.
And then we all grew a little bit older. And but it did go on for a long time. That sort of spirit of just training generation after
Yeah.
generation. As you say, it's now delightful that you look around and what you thought were your young people and are now basically in charge of the world.
Yeah.
So,
Yeah. Yeah.
yeah.
Yeah. Absolutely. Really fantastic. Did you ever regret writing SPM or essentially making an open toolbox? Because there's sometimes misuse, right? People just pressing buttons, don't understanding the more complicated things, not understanding the more complicated things. Was there ever a doubt of, I put a tool in the hands of fools, or not really?
No,
No, great.
no.
I do wonder about that sometimes with the Lead DBS toolbox, and but I also often come to the conclusion, at least on the a net positive, it is a good thing. But I have seen DCM papers that were maybe not the best ones, right? And probably if you read them, you think, oh, we enabled that tool, but yeah,
That's I see what you mean. Yes, no, certainly there is a slight sense of responsibility when people apply these procedures without a foundational training of how to best
yes,
leverage these in terms of answering their questions. But um, I tend to be quite forgiving. The majority of papers I review, I sort of treat as sort of training or PhD papers. So you're quite forgiving, and then you just try and provide reviewer comments. And the next time they do it, they'll be slightly closer
Yeah.
to the good practice that is often, and some people start to write papers about 10 simple rules for this and that, or good practice in classing
Yeah.
and Stefan very energetically trying to get people to use DCM in the right kind of way. But every everything is a learning experience, and I think you just have
Yes.
to be forgiving sometimes when people publish what their learning experiences in the open literature.
Just a few rapid fire questions, if I may. What was a true eureka moment in your career that you may remember?
Yeah, they're pretty frequent. There are several that come to mind, but I'm going to pick one which speaks to my later career in terms of theoretical neurobiology. And when I read the early accounts of um, predictive coding by people like Dana Ballard and Rajesh Rao, um, I suddenly realized it was the same objective function that was underwriting inference and learning. Um, and that to me was a bit of an eye opener that there may be one universal objective function that can explain everything. Um, and it took me back to those early days whilst in the therapeutic community in my 20s, trying to find that one explanation for in the instance associative learning, the simple explanations that would underlie a beautiful and clean and neat mechanistic mathematical psychology. So for me, that was one of many insights, which actually came from reading somebody else's paper. That was
Fantastic.
there are lots of others that
I'm
in
sure.
terms, but we'll leave it at that.
I once heard you say that active inference secretly stands for AI. Is that true or is it post hoc reasoning?
And it's I don't care. I'm too old to remember now. uh, I think I don't think it's post hoc, to be quite honest. So there are a number of ways that you could have formulated or sold or pitched the application of the free energy principle to um, active vision or active sensing, but you could have also called it sort of sentient behavior. You
Yeah.
could have also so the trick was to try and again, very much like that example about sort of the same objective function underwriting inference and learning. The active inference just turns upon the insight. It's exactly the same objective function that underwrites action and perception. And it's an objective function that lends itself to an interpretation in terms of inference and Bayesian belief updating. Um, and so you needed to have action and some kind of perceptual inference
Yeah.
in the title. So active inference was the obvious one, but I do remember thinking that's good. That's a
Because
good
it sounds
acronym.
like it's an AI. Okay. Love it.
But what actually happened was now people don't use that, so they actually write AIF for the active inference framework to disambiguate it from active inference. Now, I never came up with that. I didn't quite know what it meant when I first read it, the AIF. But now, if you read peer-reviewed material,
Just distinct it from AI, you mean?
yeah,
From the
absolutely.
okay. Yeah. Okay. I once heard a story that somebody gave you a physical Markov blanket. Is that true?
It is, and it was in fact my son, and it was a wonderful birthday present, and he got it printed up with Markov, actually, a portrait of Markov in the center of it, with keeping your states warm since 1670, whatever it was, on
Ah, wow.
on the bottom. So it now features pride of place on my on my settee, on my couch in my office at UCL. If you like, if you remind me, I can send you a picture of my son's birthday present. along
I love
with
that.
the Gothic arch from the
The
landscape
garden.
garden.
Yes, let's do that. Last question. Is there any advice you would give to young researchers entering neuroscience or academia in general today?
Yeah, I'm often asked this question, and my universal response is keep your options open. And post hoc, I've actually realized that's exactly consistent with the free energy principle in the spirit of Occam's principle and James's maximum entropy principle. But pragmatically, I think that's still the best advice. Take every opportunity you can to have a broad foundational training as possible, because at some point it's going to be useful in increasing the latitude of choice and where you end up at later life when you sort of drill down on the particular things that you're interested in. So take every chance you can, whether it's sort of master's, PhD, or just evening classes in this and that, to keep your foundational training as broad as possible. The actual advice I usually follow, though, is just do as you're told. That seems to work as well.
Okay, that's a good point. Yeah, I think in the very beginning, I actually do say that too for PhD students. Sometimes people come in and think they already have to have their own agenda, their own, ideas and so on. But it's the wiser choice often to just follow the PI for a few years and then develop these, right? Potentially.
Yeah, yeah. I mean, life is just a journey of sating curiosity at the end. And the more you can be curious without having a sort of predetermined agenda to be this or that, the better your life will be, the better the lived life will be.
Any questions I should have asked but did not ask
You should have
before we
asked about
stop?
the. No, 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 no. You've asked some wonderful questions. I'm now thinking about all the eureka moments I should have told you about, but that's not your problem. That's my problem.
Let's do
Thank
one
you.
more then. Let's do if you have time. I have time. Can you do one more eureka moment to finish off?
Um,
Or two?
uh, another eureka moment was very early on, when realizing the identity between one's treatment of stochastic processes and the differential geometry that underwrites random field theory. And this was a sort of eureka moment that Keith Worsley held my hand in terms of exposing. Um, so this won't make sense to anybody other than those people who have both a formal and intuitive understanding of Gaussian random fields and differential topology and differential topology. But just the dots suddenly linking. It's those when you say eureka moment, I'm immediately drawn to moments in my life when I realize, oh, it's as simple as that. This is the
Mhm.
same as that. They were the same thing. I just didn't see that before. So those are the kind of moments that come to mind. And in one sense, they motivate this advice to keep your options open, because if you don't spread your knowledge and your inquiries broadly, there'll never be an opportunity to join the dots because you just haven't discovered the dots that can be joined.
Yeah, yeah, yeah. Makes sense. Love it. Thank you so much, Carl. This was really a big honor for me. I know how busy you are, and again, you mentioned it's the third interview today. So thanks for the marathon. You were as bright and sharp as always, despite the two podcasts before this. So thank you. This was great.
I've enjoyed myself greatly, and I will send you those photographs, just to celebrate our conversation.
Fantastic. Thank you.